So let's start. So did you hear me?
Yeah. Hello.
Hello. So today we'll start with this new session on mathematical and physical modelization of biology. And today I will speak with Diane, who is here. She is in Paris, and she is a mathematician, so she will explain you the mathematical part of the topic. And today we are together because I will present you my biological question and how Diane helped me to answer to my question. So before we start, so for the exam, I think you are interested for the exam, usually. The first part. So you will have to present one publication selected by Diane and the other people from this week. So you will have tomorrow, yes, you will have a physician, and so you will have mathematician and physician. So paper will be selected by these people, and you will have to present you. So you will be alone when you are going to present this paper, and you will have to present this paper, and you will have to really to focus on the biological question, the model which is chosen by the author to answer to this question. Is it relevant or not? So all the questions. So I will give you more detail by email on exactly what we are expecting, what we are expecting. So don't hesitate to interrupt me, it's not clear. And also if my English is not good. So let's start. So we'll do. So I will start with. Sorry.
When will be the exam? Like in the next few weeks, or will be?
You don't have the.
No, it's on December 12th.
Yeah.
It's on December 12th.
Right, so you will have time. Do this on. Yeah, I think a student for me IT. Yeah.
I was wondering if we would have access to the presentation that you will give, like in PDF or.
Yes. Yes, I have my email, so you can send me an email. Me and one for the care student, because I don't have your email address, so I don't know. Otherwise, I can give the presentation to the other students, and you will share. But I don't have your. So it would be also better to if someone can send me an email. So to communicate, it will be easier. Because each time I go through Philippe Vallée and so again, so it's I don't know all the information.
And Jenny,
Wait.
can you share your slide for people online? Because we can't see the screen.
Yes. Yeah. Did you see my presentation?
Yeah, great. Thanks.
I didn't start. So today, I will present to you my biological question. So some of you already know the topic. So here you have my email address. So don't hesitate to send me an email, and I will share the presentation and also presentation for other people in this week. So in the lab, so I didn't put my lab. I work in a Restore Institute, so with Louis Castella and Valérie Plana. And our main question is how to regenerate in adult mammals, how can we induce regeneration in adult mammals? So after a tissue lesion or after maybe aging or this kind of process. So after an injury, so you have a biological tissue here. So here it's epithelium, recognized. You have blood vessels, here extracellular matrix, fibroblasts, neurons. So you have these different kind of cells in a tissue. After an injury, so you remove a part of this tissue. And as you know, in adult mammals, this missing part of the tissue filled with mainly with extracellular matrix. And that leads to a loss of tissue structure and then the function. So everybody knows that you have a scarring after you cut your skin, etc. So you have a scarring. And it is most frequent in adult mammals. However, in embryo and other species like Acomis mice, we put the by so here you have Acomis or MRI mice. So this kind of mammals can regenerate some tissue. And in this species, you have full regeneration. So here, as you can see, you have a full recovery of tissue architecture. It's really similar than the beginning. And so that you restore you recover your structure, then you recover your tissue function. And our goal in the lab is to know how can we have the full recovery of tissue structure in adult mammals. So here we use in the lab previously, you cannot see. Really. So, thank you. In the lab, we have shown a few years ago that adult mammals still have the ability to regenerate. So it's not completely lost. And to show that, we use adipose tissue as a model. So it's really a model. We don't aim to regenerate adipose tissue. So most of the time, people want to get rid of this adipose tissue. And we use adipose tissue as a model, and in mice, in male, in C57 black 6 mice. And so in adult tissue, the structural architecture is maintained through homeostasis and through all the life. However, after an injury, and in our case, we remove about 40% of the tissue. So you are agreed that you decrease the function since you remove a part of the tissue. So we have a loss of structure and then a function. And as I said you before, this missing part of the tissue filled with extracellular matrix, and this leads to the new state of the tissue, and we have a scar. So this architecture is completely lost, and then we have a decrease in function. However, we have shown in the lab that a treatment with an opioid receptor antagonist, but an early and present treatment with this antagonist leads to a recovery of the tissue architecture, as you can see here. So this transient treatment with the opioid receptor antagonist leads to the recovery of tissue architecture. So it is possible to regenerate in adult mammals. So it's really nice. We unlock something, but we don't know what. We unlock something that's able to regenerate in adult mammals. Yeah.
Questions?
Yeah.
If this experiment is in vivo or ex vivo?
In vivo.
Okay.
Yeah, it's an in vivo experiment. And it's convenient because the adipose tissue also for in vivo experiment is just under the skin close to the leg. So it's really easy to access. It's easier than heart, for example. And this target also with an naloxone, this target was also validated in other species. So we treat the zebrafish, which have the ability to regenerate with morphine. So morphine is an agonist of opioid receptor. And when we treat zebrafish with morphine, we stop regeneration in zebrafish. Thus, this target seems to be the same for different species. So now our question is how to recover the full tissue architecture. So what happens, how can we distinguish what is a biological mechanism that can get the tissue repair towards regeneration instead of scarring? So now for biology, though, it is our main question. But for biology, so first, we were wondering how tissue is built. So it's the first step. If we want to recover tissue architecture, we say what happens, how tissue can be built in vivo. So first, we have to go back. So a tissue, I think most of you know that, so can be distinguished in two parts. One is the parenchyma, which is the functional part of the tissue. So it is epithelial cell. In our case, it's adipocyte. You will have cardiomyocytes for heart, etc. And this part, and the other part of the organ is the mesenchyma. And the mesenchyma is support the tissue architecture, and then also it's provide also nutrient for the tissue function. So in mesenchyma, you will have extracellular matrix and also mesenchymal stem cell, I will talk later. Fibroblast, you will have also blood vessel, immune cell, and innervation. So there is already a lot of different cells which interact together, and this interaction with all cells leads to tissue function. What you learned previously in licence or otherwise, you learn simple things. In fact, we always tell you after an injury, you will have neutrophils which will be recruited, and then you will have macrophages, and this you will have switch from macrophages M1 macrophages toward M2 macrophages. And these M2 macrophages promote tissue fibrosis through stimulation of fibroblasts. So we usually, when we teach you biology, we use a linear way to present biology. But in fact, it's more complex, and it's a problem. In fact, all cells talk together, communicate together. And so here it's an example. It's mostly studied in cancer. When you have cancer cells, you will have a communication of cancer cells with all other cells in the tissue. And it was well described in cancer. It's the same for all tissue in the organism. So when we have to think about cellular communication, the problem is which communication we'll have to target, because there's lots. So you have a lot of cells, and between cells also you will have a lot of molecular and protein, etc. So biology is highly complex. And here I think you are familiar with this now. It's obtained by single cell RNA sequencing, and we look about receptor and also ligands of this receptor, and we are able to know which cell type communicate to other cell types. And here you can see all the interaction between different cells inside the tissue. So here you have fibroblast, keratinocyte, myeloid, endothelial. So everybody communicate to everybody. So this way of viewing tissue is like an ecosystem. For those who are with me in the master know that. So in the ecosystem, you have different organisms in the ecosystem. It's like all your cells inside your tissue. And the same as a communication between all these organisms, cells will compete for food. You will have also cooperation between cells, so you will have different behavior of cells. And also all these interactions are also highly dynamic in time. So it's why I hope I convince you that we teach you biology like this, the reductionist pathway. So cells which interact with one cell, and that leads to the other cell, etc., etc. So we'll have a linear way of thinking, but in fact, we are more like this. We are in what we call the system approaches. So we have to consider the tissue as a whole. and not to focus on one cell type in the tissue. And it's a different way of thinking to do the reductionist pathway approaches that you know. Here we consider the whole tissue as a whole. So we didn't focus on one species in the tissue. With these approaches, we are more interested by interaction between all the species than one species in particular. So here it's why we call emergent property. We think that tissue architecture is a result of interaction of cells inside the tissue. And the tissue architecture at the end and the tissue function emerge from this interaction. So if you change interaction, you will have at the end another tissue. The rules are the same for different tissue, but by changing interaction between cells, I will speak about extracellular matrix now, and also with extracellular matrix, you will have different tissue architecture. So for us, it's our hypothesis, and we want to focus, to think in the system approach and not a reductionist thinking. The problem with the system approaches is that, as I told you, you will have here different cells inside the tissues that interact together, and you will have intermediate entities which emerge from this interaction, and also from the interaction by this entity, you will have a global level entity. So it's only interaction. And then you will have feedback also. The global entity level will act on the cells at the end. So if we think about tissue, you will have interaction of cells inside extracellular matrix, that this interaction leads to architecture and then to tissue function, and then tissue function and architecture will act on cell behavior. So it's like a cycle. Yeah, here is why I represent. The interaction of cells lead to a tissue. Different tissue interacting together leads to an organ. And then you know about system. So different organ, for example, heart, blood vessel, like this, like the blood distribution on all over the organism. And also, you will have to think about the environment. And then the environment will impact on the organism level, then organs, tissue, and cells. So go back now to regeneration. So we think that regeneration also, the structure at the end, emerge also from interaction. And to show you, it's right, in fact. We studied, here it's a salamander. And you know that when you cut the legs of the salamander, it regenerates. And when we remove the tissue which is under regeneration from salamander, and we put in the dish, you can see that from this cell aggregate, you will have at the end a whole member here. So here it is clearly emergence of a tissue architecture from interaction between cells. The other example to show you that tissue architecture is an emergent property is organoids. And everybody knows about organoids. And what we are doing in organoids, you are doing a cell aggregate, usually stem cell aggregate. You put in the extracellular matrix with some factor, it's like cooking. And after, at the end, you will have the emergence of the tissue architecture. And cells are well organized in the tissue. And the function of the tissue results from, in fact, from this localization of the different cell types. So in fact, when you are doing organoids, it's what I told you. You will have matrix, factors, soluble factors to communicate, and also you will have cells. So in fact, tissue, if we think less complex, in fact, tissue is in fact only matrix, chemical signaling, and also cells. Because you will see for doing modeling in mathematical modeling or physical modeling, you will have to think really simple. And then I explain you why. So you really have to think what is important to build a tissue. So you only need cells, matrix, and your active factors. So behavior of cells that you know is dependent of different factors that surround these cells. So you will have chemical factors, so the soluble factor, but also the extracellular matrix. And here you can see it's the schema. So you will have cells here, which are surrounded by extracellular matrix. And you will have fluids that carry all nutrients, oxygen, etc., for cell function. So here it is the environment of these cells. And this environment of these cells will lead to the behavior of cells. In fact, when you are doing ex vivo experiments on 3D, you can clearly see that the shape of your final structure at the end is driven by the extracellular matrix. Here you have epithelium, mammary epithelial cells, and we put only this mammary epithelial cells. Here it's normal mammary epithelial cells, so you will have ducts and acini, like a mammal tissue. And here, if you put carcinoma cells, you will have like a tumor inside the extracellular matrix. But interestingly, when you put normal cells here I think it's pancreatic epithelial cells. But when you put these cells in normal epithelial cells in different matrices here it is only collagen, and here it is only matricial. So here it is collagen 4, and here it's collagen 1. At the end, you see that you have different shape at the end. And the cells are the same at the beginning, the media is the same. So only the extracellular matrix shape the final structure. So here, so if you want to simplify our tissue, in fact, you will have cells, extracellular matrix, and here it is matrix A, so speaking French. If matrix A, you will have an acini at the end, and it puts the same cell in the same media with matrix B. In our case, it was collagen 1. You will have at the end different tissue architecture. And in fact, this complex architecture at the end is only the result of an interaction between cells and extracellular matrix. And for us, this the shape of the tissue at the end can be simplified to only interaction of cells inside extracellular matrix. And if we go back to adipose tissue morphogenesis, so how adipose tissue is constructed. At the beginning, the first step is the deposition of an extracellular matrix, as you can see here. At the beginning, we only have extracellular matrix, and adipocytes still start to appear inside the extracellular matrix. So the first step here, when we look at the adipose tissue morphogenesis in vivo, the first step is the deposition of extracellular matrix. So if I summarize, it's why we have to do, as biologists before going to mathematician, and mathematician also help us to simplify the biology, in fact. So at the end, it's the simplified model. In fact, the shape of the tissue, so the final architecture of my tissue, is only the result of a mechanical interaction of cells inside the extracellular matrix. And this extracellular matrix mechanical properties is highly dynamic because it depends on the amount of extracellular matrix. And so the synthesis and also degradation, but also of fiber cross-linking between extracellular matrix components. If you link the more you have link between all your fiber here, the more it will be resistant to deformation from cells. So it was our starting point after this long discussion with Diane about what can be simplified in biology to what is our hypothesis, what is truly our hypothesis before doing the mathematical modeling. So I don't know if you have any question of this part, but what you have to know is, because you know a lot of things, you know that biology is really complex, in fact. You study all molecular pathway, cellular pathway, etc., different kind of tissue, different everything. But you will have to really simplify and to really sit down and think what is important. And the first part, and the most difficult part for biologists, it was to simplify biology. It's really hard. So no, I don't know if you have any question on biology. So Diana, you have?
Yeah, so I will share my screen.
Green. So now it's a mathematical part.
Do you see my screen?
Yeah. No, so don't hesitate if it is not clear. No, so don't hesitate to interrupt Diana.
Yeah. Okay, so thanks, Jenny. So hello, everyone. Can you hear me well?
Yeah.
Yeah, okay, good. So I'm Diane Perichard. I'm a mathematician, actually chargé de recherche at INRIA Paris in the team, former team Mamba that is called now Musclé. And so I work on the mathematical modeling of biological system, and in particular with the group of Jenny, so the Restore Lab, since 9 years now. So the subject of this course is to do an introduction to mathematical modeling in general, and in particular for biological systems, and tell you what we worked on since all these years, and how I will build as an example of a success story, let's say, where mathematical modeling has been actually useful for understanding the rules in real systems. So my part will be divided into three main components. The first one is a general introduction. So really my point here is to just convince you that math modeling can be useful in different ways to study real systems. So we will see why and how we can build some models depending on what we want to do. And the second and third part will be built on research works that we did together with Restore. The second part will be based on the mathematical model that we proposed for adipose tissue morphogenesis, calibrated and validated on the experimental results. So we will build, I will present the details of the model, the numerical simulation that enable to analyze this model, and how we could validate our modeling assumption by confronting our results to actual experimental data, and how this model has been modified afterwards to explore new questions on regeneration, as Jenny mentioned in the introduction. So we will see how the model evolved, the new numerical simulation and analysis, and what it enabled to suggest on real system. So the last part, based on what we would have learned in the second part, would be a combined in vivo and in silico experiment that enabled actually to propose a new therapeutic target to induce regeneration in mice, thanks to the prediction of the mathematical model. Okay, so let me start with so as Jenny mentioned, really don't hesitate to stop me if something is not clear. We can exchange all along this presentation. Okay, so let's start with a general introduction to mathematical modeling. The first question is why use mathematical modeling to study real systems, and in particular in biology. So there are several reasons why it can be useful. The first one, Jenny mentioned it already in her introduction, is that biological systems are complex systems that are composed of a lot of different elements, like millions or billions of different components, like cells, DNA, molecules, tissues, fibers, etc. And they interact in different ways. They can interact chemically, mechanically, via electric signals, etc. And it involves therefore these different elements also act at different scales. You can pass from the micrometer scale, for instance, if you study cell system, to meters or kilometers if you study organisms or environment scales. So there is a need, if you want to understand something about this system, there is an absolute need to simplify. This is what you do in biology. You have a question on one system, for instance, how do cell populations segregate? And it can be due to genetic factors, it can be due to environment. So you have, in a way, to simplify your system if your hypothesis is that it's mainly due to environment, for instance. Then you have to simplify to just ensure that you are not dependent on the genetics of your population. So this is something that mathematical modeling can help because it's a very precise language. And so it helps formulate precise ideas. The thing is that what you will build in your mathematical model, so in your equations, are ingredients that you define and that you control. So you have a system that is completely free of other mechanisms, the ones that you did not consider. So it's a clean system, easy to study. And it's a concise language with well-defined rules. And so you can hope to analyze it to have a theoretical framework to analyze it precisely. What you can do when your equations are well posed, is first see whether they are well posed. Then you can know if you have solutions, the form of your solutions, how they behave, like the long time behavior. You can study phase transitions. So you can have a lot of information just by using a mathematical formalism that you cannot do in real systems because you have to do some experiments that are prone and to measurements that are prone to errors, etc. So mathematics can help you to have an object that you really know and control. The last interest of using mathematical modeling is that you can perform numerical simulations. So you have your equation, you can analyze it theoretically, but you can also simulate it on the computer, visualize your solution, visualize your system as you would, for instance, look at microscopy images. You can look at your simulation, compute some quantifiers to detect the form of your structures, etc. And it enables to have a system that you can test in time very quickly. Like, for instance, the emergence of a tissue that can take years in real life, then it can be simulated if your equations are correct, quite precisely in like few hours or few minutes. So it enables to test a lot of different hypotheses and to have results way faster than in real systems. So numerical simulations are a powerful tool, as a visual tool and a prediction tool. So what objectives can you achieve with mathematical modeling? I separate them into 4 types because it would lead to different types of studies depending on what you want to do. So the first one is to understand the main mechanism at play in a system. So for instance, you have, let's go again about the question of cell segregation in population. Your question would be, what mechanisms drive 2 populations of cells to separate, right? So you will build a model based on some hypothesis. Okay, I think that it's mostly due to repulsion, for instance. And some other people would say, no, I think it's more due to adhesion. So you can build equations with the mechanical of repulsion, equations with the mechanical of adhesion, and then look at which one reproduce best your experiments. And so this is a mathematical framework that's enabled to suggest that the mechanism that you put in your model is at least sufficient to reproduce your data. So it could be, in reality, the main mechanism at play. And all the other ones, because you did not need to include them in your equation, can be seen as secondary. So this is something that mathematical formalism can help you to deduce. The second one is to test the effect of changes. Once you have a mathematical equation that or model that represents well your system, then you can change your parameters, you can change your mechanism, you can add stuff, etc., and see how the system changes. You can observe it, and so it's exactly mirroring the experiments that you do in the lab, right? You put your cells in different type of conditions, then you look at how they evolve, how the population changes, etc. You can do that also in a mathematical framework. One very important objective that is used in a lot of studies is the prediction of the behavior of your solution. This has applications, for instance, in traffic, in meteorology, in epidemiology. The objective of your mathematical model here would be to represent at least part of your data, of something that you know in your real system, and use that to predict the future. So as I mentioned before, either by numerical simulation that are time performance, so that you can see your system in a long time that you don't have access in reality, or by analysis of your solution. If you have an equation that is well posed, then you can know how your solution will evolve in time. And if your system has been validated at some part of the time, for instance, at the beginning, and represents well, then you can hope that it will predict correctly the behavior of your solution. So it's used a lot of models, for instance, for traffic flow, are used to predict the existence of jams, for instance. Or in meteorology, okay, we see that they are not very precise. It's a very complex model because there are a lot of components, but we try to use mathematical modeling to predict the weather of tomorrow or in 1 week. And the last objective that mathematical modeling can help you do is to control. This is a whole community of mathematicians that study how you can control your system in some way, either by acting on a strategic function or either by choosing the optimal parameters to drive your solution to go where you would want her to go. This has application, for instance, in robotics, where you want, for instance, the automatic cars, where you want them going from point A to point B by avoiding some obstacles, for instance. So you want to control your driving to avoid, to go in a given direction. And this you can do it by introducing some control functions in your equation. The control behavior also is very useful in medicine because it can help you to quantify the treatment that you will put in a system. So, for instance, in cancer development, where you have a treatment that is performant for killing some cells, but you have at the same time the ability for cells to develop some resistance to treatment. Then the question would be, a control question could be, how can I choose the intermittent treatment such that it's enough to kill cells, but not enough to go into the resistant state? It's a very interesting control problem that you can have in medicine. Okay. Again, don't hesitate to ask questions if you have some. So I already mentioned several fields of applications where mathematical modeling could be useful. Here it's to broaden even more the subject. Actually, mathematical models are everywhere. In every field of science, they are. You can find them with their proper equations. So here I show you, for instance, a non-exhaustive list of where mathematical modeling is used. So it's used in fluid mechanics, for instance, and in aeronautics, where here you would have equations that describe that aim to describe the motion of your fluid. And for instance, it has application in combustion, or asking how you can build your geometry of your objects in an optimal way to avoid turbulence, for instance, in aeronautics. That can be. So to answer this question, you have to model the flow to be able to describe how your fluid will evolve around your object. And so you have so the equations that are used in fluid mechanics, the most known one are Euler equation and Navier-Stokes equation. And they are, okay, we will see in more detail what they are later. In electromagnetism, here you want to describe a very different system where you want to describe, for instance, the flow of electricity. And in plasmas, so these are physical applications. Okay, so electromagnetism is Maxwell equations. And in plasma, you have yet another type of equation that describe the motion of your elements in high temperatures. And this would be Vlasov equation. Big data is also a part involving mathematical modeling, where here you are in yet another framework where you use neural networks, as we all know now. So you have neural networks, convolutional neural networks, and they can also be combined with game theory, where you have yet another mathematical framework to describe here the motion of, the evolution of your quantity. In robotics, we mentioned it already before, where you have a lot of control problems here to do. If you want to build, for instance, robots that are equilibrated mechanically. And cryptography is another example where mathematical modeling is very useful to ensure the security of your codes. This is very current now because of cybersecurity considerations. So in crypto money, of course, it's based on actual mathematical model. So these are examples in physics. And in particular in biology, you have also a lot of different systems that you can model with mathematical framework. I separated it into three main types. So you have let's say the family of systems that feature collective motion. Here, these systems have point in common. If you see from the images that I selected, I talk about collective motion, whether there are cars, whether there are pedestrians, whether there are fish, birds, etc. The common point of all these apparently very different systems is that they are composed of a lot of different agents that interact mechanically with their neighbors. So repulsion, for instance, this is a common rule of all physical systems. Say that agents cannot occupy the same place at the same time, so they would repulse. And when you have enough agents that interact in this way, very locally with their close neighbor, then you can see at a large scale the formation of coherent structures, such that the fish schooling here, where you have a population of fish that turns together, you can see that also in pedestrians, or the formation of traffic lanes or lanes in pedestrian systems, etc. So these systems here, the beauty of mathematics, what I mentioned before, is that it's a very simple framework. It doesn't matter whether what you model, what is the object that you model. It can be, for instance, if you are interested in the formation of pattern in large systems, you don't care whether you are talking about a fish or a pedestrian. What is the difference? If your mechanism is only repulsion and attraction, then you only need to know in which environment they live, but you don't care whether it's an animal, it's a person, it's a cell, it's a car. You can have the same formalism to describe the motion. So this equation here, you can find Köcker-Smell model, the Friesen model, etc. This is an example of a model that works really, really well and is very simple and that can explain different patterning in birds or fish or pedestrians, is the free zone model. It's a model that is very simple. It describes the motion of each individual through three basic mechanisms. On the short scale, it features repulsion. It just says that two people will repulse up until a certain distance. In large distance from one individual, it will interact with the other via attraction. That means that one individual would be attracted to a group of individuals, want to go towards the group. And in an average distance from you, then you suppose that you align. If you have someone that goes in the direction, you arrive like this, then you would tend to adopt the same speed. These three very simple mechanisms, so they are encompassed in what we call the free zone model because of this free zone. They can explain a lot of different patterns. They can explain, for instance, the formation of flocks or whatever. And they also show that humans, when there are enough of them, know better than fish or sheep. They are just basically interacting mechanically with neighbors. Okay, in biological tissues, as Jenny introduced them, you have different type of systems, so they are not necessarily self-propelled. So here in collective motion, you have individuals that can move in one direction. You have in biological systems, for instance, cells that are passive, that just move because of the forces that are acting on them. And you have different type of cells, fibers, etc. So here in biological tissue in general, you can be interested in describing the blood flow. Here you would join the fluid mechanics community that we talked before. You can be interested in how cells move in the medium, depending on the environment they are evolving to, or how tissue wound. Actually, this is the question that interests us here. And in medicine, you can have also different questions where mathematical model can help you. Is, as I mentioned before, how to optimize cancer treatments when you have a molecule, for instance, that can act on some cells. And you can also work on neuroscience, for instance, to understand how the connectivity of your brain can lead to degenerative diseases, etc. Okay, so these are some examples. Of course, there are way more. It was just to illustrate different questions that you can have depending on your system. So let's say that we want to use a mathematical framework to explore some hypotheses about some system. They cannot be as much more general than that. What model then could we propose? There are a lot of them, as you saw from what I mentioned already before. So how to choose the right model? You can ask yourself 3 questions. At which scale you want to look at your system? Scale meaning, do you want to have a precise description of the motion of each individual in your population, or do you know, or your object is a continuous quantity, such like, for instance, the concentration of a chemical of something? You don't care where the molecules of your chemical are, you just look at the concentration. That is a macroscopic quantity, a continuous quantity. So depending on the scale, that would help you decide whether you would use a discrete or a continuous model. Then you can ask yourself, what is the importance of statistical fluctuations in your, if you consider that your system is mostly driven by statistical fluctuation, you will have all interest to include that in your mathematical framework. And so you would prefer to use a stochastic model. While if what you are interested about is an average behavior of a population that is deterministic, for instance, if I look at the motion of a ball that I push, then it's a deterministic motion. Thanks to Newton's law, I know that if I exert some force in an object, it will move off some quantity that I know. So at this scale here, There are some statistical fluctuation if you look at the molecules of air that interact with your balloon, but you can ignore them because they are very small compared to the motion. So in this case, you can use, you can justify to use a deterministic model, but the motion is very precisely described. And then the first thing to ask is the level of understanding of your system. Whether you want to have precise rules in the interaction that you put in your model between your elements, or whether like classical laws are sufficient, like this, for instance, the Kleber's law for metabolism. Do you want to understand precisely, to model precisely all the interaction leading to the consumption of energy by your cell or organism, or whether you can say that, okay, macroscopically, it evolves like the volume at the puissance 3/4. That's enough for you. And so this helps you to know which rule you will put in your model. So it's just to say, okay, which type of model can I use to, so you see that it's driven by the questions that you have on your system. And it must be adapted to the scale at which you want to look at your system. And how do you decide your scale? Well, it depends on your observable, on what you can do on your system to test it. Okay, the very important stuff is scale. So this is one of my research interest. So we already talked about systems that are composed of agents, for which we describe precisely the motion of each. Okay, these are models that are called at the microscopic scale. So micro is not micrometer. Okay. Micro can be 1 pedestrian, for instance. It's just when I talk about microscopic scale, I talk about the scale where I see each agent. So for instance, here, I show you a picture here on the right part. It's a picture of two cell population, one stained in green and one stained in red. But you see, in a Petri dish, okay, just part of the Petri dish. And you see that you cannot see actually the elements. What you see is only patterns of colors, green or red, but you don't know that they are cells. If you zoom in, then you can start to see the cells. This is what I call microscopic scale. I could have done that on a crowd of pedestrians, I don't care. So if you are at a scale where you can follow the motion of your agents, then you have all interest to use an agent-based model. That is a model that will describe the motion of each of your elements. Not only the motion, it can describe quantities that are proper to your agent. For instance, the direction of velocity, the position, the temperature of each, I don't care. The advantage of using this kind of models, what we call agent-based model, is that they are very precise in terms of modeling, because you describe each interaction between each agent. And so you can hope, because of this modeling precision, you can hope to have a link with experimental data. As a drawback, you can imagine that they are computationally challenging, because when you study systems of millions of interactions, you have millions of equations to solve. So numerically, it's very costly. And the problem is that you have to resort to numerical simulations, because you lack of a theoretical framework to analyze systematically your solution. So you have to do simulation to understand how the parameters, how the mechanism you put in your equation impacts your solution. And the simulations can be costly. So when you are interested in the study of this scale here, the one on the right, then you would prefer to use continuous models that describe mean variables. Here you don't care whether exactly precisely where each of your agents are. What you're interested in is the average number of people around some point in space. That is what we call the density. You can also have macroscopic quantities such as temperature, pressure, mean orientation of your agents, etc. So in this case, these mathematical models are called continuous models. And if they are simple enough, you can hope to have a theoretical framework to analyze your solution analytically. Not necessarily analytically, but at least have some understanding on the behavior of your solutions, for instance. And they are computationally efficient because they enable to model systems composed of whatever, millions, billions of individuals, because they just describe this average quantity. The problem with this model is that because you zoom out and you look only at the behavior of the mean, then you lose the precision of your modeling. So one of my research activity, very active, is to make the link between these two descriptions. This really mathematical consideration that is very important because it would enable to build macroscopic models that are actually derived from a microscopic dynamics, and then you gain the modeling precision of the agent-based model. Okay, so I mentioned already two types of mathematical model. Actually, we can group them into three main types. The type of equations that you can use that you will have if you use a mathematical model are of three types. You have the first family is called ordinary differential equation, ODE. And as an example to illustrate what is an ODE, I use the SIR model that we all know since COVID, at least. And so this is a very basic model of epidemiology that describe the population of humans, for instance, that is divided into three compartments. You want to describe the compartment of the sensitive people who can get infected. This is this quantity S, so NS would be related to the number of sensitive people in your population at time t. Then you describe the other compartment of the infected people, the one who got the disease, described by this Ni of t. And the last compartment is the recovered people. So either you count the number of deaths or the number of recovery. Okay, in this model, you see that you have three equations that describe the evolution of these continuous quantities that depend only on time. Okay, and so what you can observe is that here the mechanisms that you put in the SIR model is to say that an infected people, I mean, you will create new infected people if a sensitive people is in contact with an infected people, okay? You will become infected with a rate beta. And then infected people can recover with a rate gamma. So that's why the evolution of your population of recovered people increases with a rate gamma and depending on the infected people here. And the sensitive people, they decrease in your population because they got infected. So actually this model here, that is a ODE system in the sense that you only have one derivative, you only have one variable, so one derivative in time here. It can be derived from a chemical reaction network where you see one molecule of sensitive, so one sensitive person interacting with one infected creates with a rate beta with some probability an infected people. And an infected can recover with rate gamma. This leads to this system of equations. So you see that here, your continuous quantity NS of t, for instance, does not depend on space. So this is a model for a population where everyone can interact with everyone if you want. So this is one limitation. There are a lot of limitations. It's a very simplified model for epidemiology, but that enabled, for instance, to define the basic reproduction number, you know, this R0 that we heard so much about during COVID. This comes actually from this framework here. That is a quantity that can predict the fact whether your population will increase, like you will have an epidemic, or whether it will die out in long time. This is the analysis of this model that enables to predict actually where you will go. So, but one of the limitations, for instance, is that you don't take into account the spatial configuration of your people. Means that here you cannot use this model if you have people living in France and people living in Russia, because in this model, they are supposed to be, you don't care where they are, they can interact. So when you want to account for spatial effects, then you have to resort to other type of model. The second big family of mathematical equations are what's called stochastic differential equations. Here, these are stochastic in the sense that, so if you remember one of the questions that we had to decide on which model we use, is where in a system where statistical fluctuations are important. I mean, you suppose that they are important in your motion. So in this case, you use this type of equation. They are a bit peculiar because it's not a quantity. So here, let's say that this is the equation of motion of xt. That is the position of a particle. The thing with SDE is that you have implemented in your model the fact that you have a stochastic process. That means that it's not a quantity that is known, it's a random variable. So actually, here in this example, your stochastic process xt evolves randomly around some value. This equation just means that if you are in a position xt, the motion in between two time step delta t would be normally distributed around mu delta t and with variance d. That means that you just know, if you are a particle at time t, you just know that you have a probability to move in direction mu, and but you can also do a random jump around your position. So that's very true in biological system. You know that the cells do not exactly move in some direction. They can also move randomly around their position. Okay, so these are SDE. But when statistical fluctuations can be neglected, then you can use deterministic models, and these are modeled by PDs. I mean, ODs are also deterministic motion here. You see that you don't have random variables. You know, if your population is at some value at one time, you know exactly which value it will take at the other time. But PDEs, the difference with ODEs is that they have several variables. So for instance, they can work here. I work on a quantity that I call C, which depends on space x and time t. And that can describe, for instance, the spatial density of particles. So the average number of particles I can expect around point x at time t. Here I give an example of a PDE that models the convection diffusion equation, or advection diffusion, or again, transport diffusion equation. So I just actually describe the evolution in time of this quantity C around point X. It will decrease by a quantity mu in the direction of its derivative. That's just a transport term to say that your population is transported with rate mu, and it will diffuse with coefficient d. Actually, this equation, you can obtain it from the limit of a large number of people interacting via the SDE process described before. If you have a lot of people that have a probability to go to be transported by mu, and some probability to move randomly around their position, that leads to a transport diffusion equation. Okay, this was just to give you some examples of equations that we can have. And now I will give you this is the stages of modeling when you want to apply mathematical modeling to an actual biological problem that you have. So usually you start from some system and a question on your systems, okay? So it's exactly the same principle as if you have a research question on your biological system. So we share this initially, and the first thing to do is to decide on heuristic rules. So you made some hypothesis to drive your question here. This helps you, these heuristic rules, then you implement them into a mathematical model. I will show you how, for instance, the question can, or hypothesis on mechanisms can translate into some equations. So this is a very closely interdisciplinary work that you have to do together with the biologists to decide on which rule you implement and which one you neglect and you don't put in the model. Then once you have built your mathematical model, you can do several stuff. So you can numerically simulate, for instance, your model and start to test and validate your hypothesis by, for instance, by playing on the parameters in different regime and see whether you reproduce your biological reality. So you confront your model with some actual elements, and this enables you to tune your mechanisms, add some other that are necessary, or remove some other that were not well chosen. Then you can go deep down in the analysis of your mathematical model. So for instance, if you started from an agent-based model, you can question what is the continuous formalism associated with this agent-based model that enables you, once you have a continuum model, to analyze it systematically and to have some information, precise information of your mechanisms on your solutions. Or you can do sensitivity analysis. So you do a lot of simulation and then you look at you try to detect all the behavior that your model can have as function of the parameters and see which ones reproduce the reality, which ones not, etc. This whole process enables you to build some mathematical model that is validated on experimental data, and then you can use it. The last test is to use it for prediction and control, as we mentioned before. These models, because they are simpler, faster than experiments, then they can help you. They can drive actually new experiments. They can lead to clinical trials, to tests, etc. This can be a very powerful tool if it has been before well calibrated and validated on your data. Okay, I hope I convinced you that it's not completely crazy to try to develop a mathematical framework to answer a real question. And so, okay, maybe you have some questions on this part before going on.
Okay. So the first part between biological reality and the building of mathematical model it takes time, and it's mostly discussion about the biologists and also mathematicians. Because is it okay for you the different kind of model you have and the limitation? Because most of the time it's the choice of the model common from this discussion between mathematician and biologists. So you will see as Diane will show you my agent-based model. You can complete that, Diane. It's very precise. So for us, as I told you, an agent for biology is just one cell type, for example, matrix. So it's different parts of the model. And so the agent-based model is really time-consuming because you focus on one agent. That's why I focus on one of you, your property and how you behave and like this. So it takes time to analyze each component. So this one is, but it gives you a lot of information because I know all of you. Not really, but. And the other one, when you are doing two continuous models, the macroscopic scale, is I take all the group of. And so when you are doing agent-based model, it's sometimes it's what we are doing and try to do to start because we are doing agent-based modeling and to go on the continuous model. Because, for example, we are only study regeneration at one time point. So only 2 months for the tissue. But if you want to follow the whole life of the people, we have to go through a continuous model. You cannot, so it's why, and this interaction between agent-based model and the continuous model. I don't know. It's okay for you, Jan.
Yeah.
So each model has its limitation, and I don't know if it's clear for you, the limitation of each model. Yes.
So in that, we usually like combine different models together. Like you use, for example, the agent-based model, and then you continuous model at the same, like, for example, experiment, I would say.
For us, we start with agent-based models. And now we know really well this agent-based model. We know its behavior, what is important or not. So we can go on a continuous model. Because you have to. You cannot focus on one for each agent. So you will have to know really well the rules before going through a continuous model. So it's why we start by using this agent-based model. And sometimes also the first model that is built is not really good, and so we go back and so it takes time, don't worry. But yes, for you, when you are reading a paper on mathematical modeling and so on, you will have, as Jan show you, you have ODE and the explain, so you don't have to retain all the formula. But to know why people use this model instead of another one to answer to this biological question. For you to, or the goal of this teaching. That you can go, yeah.
Yeah, and so one very important remark that Jenny said, so I will just jump on that also. So mathematical model enable you to simplify your system, as you mentioned, but it obliges you to simplify your system, because the point of using mathematics is to be able to analyze it. So you have to have a simplified framework. You really have to keep in mind that as much detail you put in the mathematical model, it's gets harder and harder to be able to predict anything. It's the problem of, for instance, over-parameterizing your problem. So, like, for instance, you have a linear curve, and then you do a complex model with 5 parameters. To interpolate a linear curve, you just need 2 parameters. So if you have 5, that means that you will have an over-parameterization, and you will not be able to use that for prediction, because you have a whole set of parameters that are useless, and so that are not used in your prediction. So it's really the point of using mathematics is really to come up with a minimal model, a simplified model, as much as possible, so that you can have an object that you can manipulate precisely. So yeah, it's actually it's actually a requirement. The hardest part for biologists is to accept to neglect some phenomena, and just to say, okay, I will not go through every differentiation pathway or whatever. I will just consider. So you will see that the model that we propose, okay, I will jump right into that, and so don't be afraid about the simplicity of the framework. So okay, so in particular with this work, our objective, so the first objective was to understand the mechanisms involved in tissue morphogenesis. So how a tissue architecture emerges from nothing, or from embryonic to an adult stage, and how it equilibrates to this architecture. So we were really interested in the mechanics and the emergence of forms in adult tissue. So we used, as Jenny mentioned initially, adipose tissue as a model, so model in the biology sense. And we started from observation as we always do. So here I show two microscopic images of adipose tissue of adult mice. So the left figure here depicts the whole tissue, so what we can call the macroscopic scale, because we don't see the individual cells here. And here on the right part, zoom in the structures here, and they are stained according to their type of element. So in particular, adipose tissue, you probably know way better than me that is composed of different elements. So the differentiated cells are called adipocytes. They are stained in red here. And they are organized in the adult state in what is called lobules. So these like rugby ball type of shapes. That are comprised in a more or less organized fiber network of collagen fibers that are stained in blue here. And finally, the last thing we see is stem cells, if I don't mistake, so undifferentiated cells that can become specialized and become adipocytes later. So what we see here is that the system produces these very specific structures of lobules composed of cells of different sizes. You have big and small cells in a matrix. If we look at the organization of these lobules, so we can see the contour of the lobules here in the whole tissue. And more specifically, we can see that depending on the part of the tissue we are looking at, we have different kind of organizations. We are on this image side, regions where the lobules are round, quite more or less round and well defined. We have regions where the lobules are very elongated, and we have zones where the lobules are not really organized, and it's less structured. So we have these different levels of organization. So the question was whether we could use mathematical model to explain, or at least to reproduce this, the emergence of these structures. So starting from this observation, we proposed a very simple model that featured only two elements, the cells that are supposed to be modeled as 2D spheres. So we propose first a model in 2D, okay? So cells are represented by a center, xi, and a radius ri that can depend on time to model cell growth. And fibers, they are the other elements, so they are represented as a center yf, an orientation angle theta f, and they are segments of fixed length. So we have these two family of mathematical objects. And then we have to describe some biological phenomena, so the ingredients that we put in our model. So again, we want to select a minimal amount of biological phenomena to reproduce our structures. So in order to model the fiber network, we consider that two segments can link randomly with some probability and create an attach point that is called K here. So two segments that cross each other can create a link, and this link can disappear also randomly with some parameter, some frequency. Then the other biological phenomena is the insemination of new cells. So as you see, we only have two elements, the adipocyte cells and the collagen fibers. Yes, sorry, I didn't mention. We consider creation of links enabled in order to model long fibers as a set of connected segments. So a segment is not one biological fiber, is already composed a bundle of several small fibers, but we can have with this model even longer fibers by connecting them one after the other. So this is why we introduce this phenomena. So the insemination of new cell is just modeled by the random apparition of one cell of minimal radius R. And this has been introduced to model the differentiation of stem cells into functional adipocyte cells that can stock lipids. Okay, so we don't model stem cell. We suppose that there is a pool of stem cells that is in the environment. We don't need to model them, and they just randomly differentiate by making an adipocyte of minimal radius here. This is an important hypothesis so that we put here, is that stem cells don't have any mechanical impact on the tissue. So we can just neglect them. They are just behind the insemination of new cells. And then we model lipid storage, so the stock of lipid by the cells by making the cell grow from a minimal radius to a maximal radius. That is a parameter of the model. So this is cell growth. These are the only three biological phenomena that we put here, but you see that behind they represent some other hypotheses. So the important thing when you build a mathematical model is to know what do your hypothesis encompass actually in the real world. Then we describe the mechanical interactions between our elements. So the first thing that we consider, it's based on the observation. Actually, we see at the adult state, cells and fibers are segregated. You have lobules of cells and the fibers are around. So we suppose that cells and fibers repulse each other. So this, I will explain how we model that, but this is the first mechanical consideration. It's repulsion between cells and fibers. Then we suppose that fibers that are cross-linked are submitted to an alignment potential, so they tend to want to be aligned together. This is a mechanism that we introduced in order to give some rigidity to our network, some strength. So you can imagine that if two fibers are linked like that, and then you have a cell that grows below, then it will push on the fiber and it will make them then rotate one from the other. And so the mechanism of alignment was a way to give our fiber a feedback mechanism to push on the cell because they want to align together. Okay, so this is a mechanism that we imagine to give some strength to our network. And finally, the last mechanical element of the model is a very naive way to take into account the fact that fat is an incompressible fluid. So we suppose that cells cannot overlap. Okay, so this is a constraint. Cells can move, but they cannot overlap. Okay, so based on these ingredients, then we can describe the motion of the position of our cells and position of our fiber and rotation of our fiber by following a physical principle. And the physical principle that we chose here was to say that at any time, the system will be at mechanical equilibrium. This could, we could have another rule for motion by using, for instance, Newton's equation of motion. Mass times acceleration equals sum of the forces. Because here we have the forces, and we could have used this framework. Here we are in a particular regime where we say that people, so the cells and fibers, will instantaneously mechanically equilibrate in order to minimize some interaction potential. So the interaction potential, this functional W that depends on the position of cells, position of fibers, and orientation of the fibers, will be minimized at every discrete time. And it's minimized under constraint, hard constraints, that cells cannot overlap, this functional here, and that fiber crosslinks must be maintained. So what does it mean? This, how does this give you motion of people? You can think about it as a discrete time process. You have an initial configuration. So you have, for instance, fibers and cells. They are positioned in some part of your domain. And so at in between two discrete times, then some biological phenomena will appear. So you will, for instance, introduce some cells, you will make cell grow. And so that given two cells, if they grow, then you can create some overlap. And so they will move in order to avoid overlapping and to repulse with the other ones. So this gives you motion in between two time steps. So how do we model that? We'll just to make things a bit more clear. So how do we solve that? Sorry. In between two time steps, you see that we have a minimization algorithm to solve. Means that given a configuration and some biological phenomena, I need to find a position that minimize my equilibrium, mechanical equilibrium. So we use this functional W, as I mentioned before. This is the total mechanical potential. And it's comprised of the cell fiber repulsion potential here and the fiber alignment here. So how do we model cell fiber repulsion? If here I draw a fiber, okay, with a position YF, then we suppose that each fiber generates a potential, the isolines of which are ellipses here. The isolines, that means that the potential is non-zero inside the ellipse and zero outside. So if you have a cell here, then the potential felt by the cell will be positive. And you want to minimize this potential, so the cell will move in order to go outside of this ellipse. This is how we model repulsion. In the same way, if the cell is here, the fiber sends a positive potential due to the presence of cell, so it will rotate so that to orient our ellipse such that the cell goes outside of this potential. So you see that this can be easily described by just a function that has a support in the form of ellipse around the fiber here. And the gradient of and the motion of your cells and fiber will just follow the gradient of your potential in order for them to separate. So this is repulsion here. Then fiber alignment. How do you put that in equation? Again, you want to define a potential that is positive if the fibers are not aligned and that is minimal when the fibers are aligned. So what we use as a functional is the square of the sinus of the difference between the angles of your fibers. So you see if you have fiber I that is connected to fiber J, if you look at this function here, sinus square of the theta I minus theta J, it is zero exactly when theta I equals theta J. That means that the two fibers have the same orientation angle, that means that they are aligned, and it's maximal when they are perpendicular. Okay, so if you want to minimize this functional, that amounts to align your theta, your orientation angle. Okay, then the exclusion constraint, it's the same. You express it as a function of your position. You have to express all your mechanism as function of your positions, okay? So here, if you want to have a functional that is positive when people are overlapping, you can just look at the function of the sum of the radius square minus the distance of their center. You see that this function is positive if the two centers are closer than the sum of their radius. So this gives you also a function, if you minimize it, that it would become zero when your cells do not overlap. And finally, you do the same for the fibers. You want to prevent them to go far from each other. You want to make them attach at their attachment point. So you define a functional that is positive when the two attachment points are not at the same position. So it's all a matter, as you see, of transforming your rules, your mechanical quantities into functions of position. And then you know how to describe the motion of your position and angles in order to minimize a function. Okay, so you see that the model is quite simple in terms of mechanisms. We don't have so much stuff into account. We have cells that appear, that grow. We have a fiber network that is composed of segments that are connected dynamically in time to their crossing neighbors, and we have alignment of these fibers. So fibers, really, the important stuff is that the links create constraints on their motion. That means that two fibers that are linked, they can move, but they have to move together and maintain their attachment site during their motion. Do you have questions on the model? Is it clear? Okay. Don't hesitate if you have questions. Also online, you can put in the chat if you have questions, because it's open here. Okay, so I described the functionals that we defined for the mechanical interactions. Then just to tell you how we model the biological phenomena. So as I mentioned, cell growth is supposed to be a regular process. This is an hypothesis, a simplification hypothesis that we put. So cells just grow regularly with a linear volumic growth. So just their volume increase some quantity that is given up until a maximum size. That is also a parameter. So this is a deterministic process for cell growth. It's an oversimplification of the system. You can imagine that adipose tissue do not just stock regularly energy, but okay, this was just to see whether we could reproduce architectures. So we postulate this law. Then the appearance of new cell that model stem cell differentiation. Here it's supposed to be a random process. So we model it with the Poisson process. That means that you just have some probabilities that cells will appear in between two time steps. And the question is where they appear. So we have explored two possibilities. The first one is that stem cells, adipose cells appear randomly in the domain. So it's a non-biased process. Or we also studied whether cell could appear close to existing cells, existing adipocytes. And this enables to question the presence of a subjacent vascular network. If there is a vascular network in our environment, then that means that you have preferred location where cells would appear, close to the blood vessels, for instance. So the way to test that in the model was to test two ways of making cells appear, either randomly in the domain, supposing that there is nutrient everywhere, either biased, like giving a higher probability for cells to appear next to existing cells. We will see what it enabled us to conclude here. And finally, the last biological phenomena is creation and suppression of fiber crosslinks. So this is also supposed to be a random process with frequencies nu f for creating and nu d for deleting. Okay, so you just have some probabilities. And there is a quantity that we know that is related to the number of fiber links that we have at the equilibrium of this process. This quantity K-L that will be very important later. Okay, so now I have described all the ingredients that we put in our model. I hope it's clear how we solve, at least numerically, these equations. And if it's not, in any case, there are numerical simulations. So now I will show you what the model is able, what this very simplified model is able to do. So, of course, when the model description is not complete, it's not complete because I didn't talk about the environment. I didn't talk about the domain in which we evolve. You see, I talked about position of some cells, position of some fibers, but I have in my mathematical framework to say, where do these mathematical objects live? So I mentioned quickly that we are in 2D. So this is an image of an initial condition. We are in 2D and in a domain with periodic boundary condition. What does that mean? That means that every people who go out, for instance, from the left side, will re-enter in the right side. So basically, it's saying that we live on the 2D torus, and that enables to model an infinite part of a tissue. There is no boundary, it's just a visual effect, but these two borders here are the same, okay? So we have boundary conditions everywhere, and our initial condition is just a network of fibers that we randomly throw in this domain. So we just throw n fibers, n being the parameter, with random orientation also. And then we just link them with some probability also. So it gives us an initial network. Suppose we start from an initial network. And then we let the system evolve. So remember that as a biological phenomena, there is stem cell differentiation. And here we already are at some time in the simulation. So you see that cells have appeared here with minimal radius. So cells will appear. So cells appear in the domain. So we have also fiber aligned. We just send you. We have fiber that align because of mechanical alignment. We have links that appear and disappear. So here in this simulation, the dynamics of the fiber network is very low. So the links at the beginning stay quite a bit during the simulation. So then you see that because of stem cell insemination, you create some cells up until the maximum number is reached. And what we observe in this simulation is that we have the spontaneous reorganization of the system in form of clusters of cells that are living inside a fiber network here. So we didn't put anything to force the cell and fiber to adopt this configuration. It's just a result of the mechanical interaction that we consider. Because the fiber are anisotropic, that means that they give some information of their orientation to the cells. Then the cell will be packed into some clusters. And this cluster, in this simulation in particular, have different shapes, and they are more or less ordered. This is a simulation that you saw the film. Hein, Jenny?
Yes, we can put the fiber.
You saw the fibers move or just the images? No, okay. I just had a doubt. So this was a simulation where the cross-link process, so creation and suppression of links, was quite small. Now, I will show a simulation where here you have a very fast dynamics of creating and suppressing the links between the fibers, and you have a high number of links. In this case, what we see is that quite different behavior than before. Then the links, the fibers, because they unlink. Then remember, in the first one, you have configuration of these fibers that were untangled, and because the cross-link were maintained in time, then they cannot align. Here, you break, sorry, the configuration, and so the fibers are able to align with their close neighbor, and you see that we create this bundle of a lot of fibers that are very aligned and highly linked. And as a consequence, it transmits the alignment of this network here, transmit to the alignment of the cell population, and we see that we do an architecture that is very different in this tissue compared to the one before. So you see that playing on the parameters enables you to have different type of configuration in your cell and fiber population. You can have simulation where you have more or less cell cluster that they are more or less aligned, and two different type of clusters can coexist inside the same network. So we already identified the number of link and the frequency of linking and linking as parameters, as parameter controlling, sorry, the different architecture. But the beauty of having a mathematical framework is that you can quantify that. And you can question what is the influence of each parameter of your model. So actually, we did a thorough parametric analysis by sending a lot of simulation with different parameters values. And then we questioned what type of architecture we had at the end as function of the parameters. So to do that, see, in a quantitative way, we need to have quantifiers, right? So what interested us was the shape of the cell clusters, so that we quantified through this elongation parameter E, quantifier E, sorry, and the alignment of the network. And this is this quantifier A. When this quantifier E, it gives a measure of the alignment. If it gets larger, that means that the cell clusters are more aligned. And the A is the same, but it's the alignment of the fiber. As larger as the A becomes, means that the fiber clusters are aligned. And these quantifiers, we can plot their value as function of the value of the parameters. And this enables to detect phase transitions. Actually, we saw with this model a sharp phase transition controlled by the parameter nu-d. This is the unlinking frequency of the fibers. So this is a measure, the parameter nu-d is a measure of the remodeling speed of the network. So if we have networks that have low remodeling speed, that means that they create matrices that are lowly aligned, as we can imagine, because we have a lot of zones where the fibers are entangled and they are blocked in mechanical configuration. While if you increase the remodeling, this enables to reorganize your fiber to have a very aligned network. And there is a critical value of remodeling that pass from disorganized to organized network. And we see that if we look at the elongation of the cluster, we follow exactly this behavior too. So it means that the fiber network alignment transmits to the cell cluster alignment. So, okay, here I focus with this analysis, we could detect that the different architectures were mostly controlled by the parameter, the remodeling speed, and by the number of links of the network. So that was the phase of analysis of the model. So all the other parameters are actually secondary. The main ones that control the structures are the ones that are linked to the fiber network. That was already a result, a very interesting result. The other stuff that we did, okay, so I think I don't talk about it here, so I will just mention it. Here it's in the case where cell insemination is uniform in the domain. Cells can appear anywhere with the same probability. We tried also to make cells appear more probably close to existing cells, and we showed that it does not impact the structure at all in infinite. If we look at a simulation, we can make sense of that. At a point, so initially we have some clusters that start to form, but they are not defined. And then the fiber and cell will reorganize, and this you will have families of cells that will regroup. For instance, if you look at these two zones here, these two cells, they will regroup in time because the fiber will disappear, and then it will create one unique cluster. And so actually, if the cells would have appeared more probably here where there is already people, it would not have changed the number of clusters here. This is what the model proposed. It's because of the reorganization of the fibers. In any case, you will have segregation of the two families. And so what does that suggest in terms of biology is that vasculature could be secondary to organization, architecture organization, because it's not necessary to reproduce the structure. But to be able to have this conclusion, we need to show that our model produces structures that are close. So now we go to the validation of the models with experiments. So we have some quantifiers on the structures of the mathematical model, and our goal was to compute the same quantifiers on the microscopy image of real tissues. So what we did was to develop automatic segmentation techniques from the microscopy image. So this is an example of an image of adipose tissue where you have the membrane of the cell that is stained in green here. So you don't have the fibers, it's in the black background, but you see the contour of the cells. And so there is an automatic segmentation algorithm that enables you to automatically detect the presence of the cells. So we developed this tool. Given an image, we know where each cell is. We have the position and size. And then we did develop a semi-automatic tool that, given an image, it detects automatically the clusters of the cell. So the big structures that we see here. This tool is semi-automatic in the sense that sometimes it does not, it has a hard time to distinguish between two lobules, but the eye also has a hard time, and so we have to help him by drawing lines saying, okay, this I would like to separate them. So, okay, this is just I take this moment to drive you to think in your future research, in your future work, when you have data, then you have all interest to try to look whether there are automatic segmentation too. Because I know that it's not necessarily a reflex, and there are some theses that are dedicated into doing this, what the algorithm do by hand. So you have people who work on, you can do it by hand, right? You can say, okay, you click on the inside of the cell by eye, etc. But the power of doing this automatic tool, that you gain a lot of time, and there are a lot of softwares that can enable you to do that. And you can go to mathematicians if you want to be lazy and to gain some time for automatic computation. So think about using segmentation tools to help the analysis of your data. So, okay, so these two tools enabled us, given an image, to have the information of all the position of the cells and of the number and form of the lobules. And so we could compute the same quantifiers as we had in our simulation. And so compare quantitatively the model the architecture of the simulation results with the architecture of the microscopy image. So how do we do that? We have, for each image here, we compute our morphological quantifiers, and then we look in our database of simulation, which one is closest. And we could, for different images, for 5 or 6 different images, we had at least one simulation that reproduced the same features. Okay. Be careful here. We are, of course, we are not saying that it's not like an exact reproduction of the system. We are not saying that we have a model that reproduces exactly our tissue. We have a model that reproduces something that is close. But if we look more precisely, you see that our lobules, okay, this one has the same, the close shape as this one, yes, but it has way less cells. Our cells are way bigger than the real adipocytes. But it's not the point. Actually, the point is that the model suggests that it's mechanics that could drive the stuff, even if we are not exactly at the same scale. So one cell can represent 10 real adipocytes. The thing is that we simplify the system. And we look at the global picture. But of course, this last consideration made the fact that we published in a journal of theoretical biology and not in Nature, for instance, right? We just propose a simplified mathematical framework that the only message that it gives is that it could be due to, I think it's the title, yes. Our title is Simple Mechanical Cues Could Explain Adipose Tissue Morphology. Right? We don't have the pretension now to have a digital twin or whatever. At this stage, it's just a model that reproduces some architecture. But as an important message, is that it's a first proof of concept that vasculature could be secondary to organization. And this actually is a very interesting stuff that the model proposed, because before what is mostly thought of in the biological community is that, and as Jenny introduced it in her talk, is that the tissue evolves in 3 steps. The first is the creation of a matrix, then the apparition of a vascular network, and then the apparition of the adipocytes. So what is thought of is that vasculature is essential to the organization of the adipose tissue. We're not saying that vasculature does not exist, but that means that it's not the one that drives the architecture. What drives the architecture is, for us, the mechanical interaction between the cell and the extracellular matrix. Okay, it's okay for now?
Okay.
Okay. So based on this model, we were quite happy about this information that we could gather on this simple system. And we took a step further and asked whether we could use this model in a different setting and in the context of, in particular, in the context of wound healing and regeneration. Could we use the same framework to question the mechanisms in this after injury? So, Jenny, you want to do this biological?
I don't know. I think most of you know about inflammation or the process, more or less. Yeah, I think.
Yeah.
Okay. It's okay.
Okay, perfect. It's just a sketch. Remember what you already know, and I will not. I'm a mathematician, so I will not dare to. But okay, there are 3 phases after injury: inflammation, proliferation, and remodeling. So, so we had to modify the model in order to take into account the reconstruction phase after injury. So our main hypothesis that we wanted to explore here with this mathematical model was whether regeneration could mirror development. And how we can ask that is that whether our simple model of tissue emergence could also serve with the exact same mechanism to explain regeneration or wound healing. And this is like a very interesting question, right? Is regeneration, reconstruction after injury, very different process, or is it the same rules as creation of a tissue? So to explore that, we used our previous model. Okay, so this is an example of simulation at equilibrium, and we model an injury exactly as the biologists do in the lab by cutting part of the tissue. So we just remove all the components that are in between two positions in our tissue. Then we had to modify the model. So as you remember, our model did not incorporate fiber insemination. We had cell insemination, but not fiber insemination, because we started from an already existing ECM matrix. So now for injury, we have to reconstruct the whole stuff. We have to reconstruct fiber network and the cells. So we modify the model. And in order to take into account inflammation, which is something present in real tissue, we consider the production of a chemical signal, okay, that is just described by its concentration or density that depends on space and time. And here you recognize probably, I hope, the transport diffusion equation that we saw before. So basically, this signal is produced where there are gradients in our system. So gradients, that means that it's positioned in space where you have a difference of concentration of agents before and after. So here, for instance, when you cut here, you have tissue on the left part and no tissue on the right. So there is high gradient here. While if you are positioned at this spatial position here, you have tissue everywhere, and so you have 0 gradient. And this is like a very interesting question, right? Is regeneration, reconstruction after injury very different process, or is it the same rules as creation of a tissue? So Okay, so the presence of gradients, it just triggers the creation of a chemical signal, and this signal then is supposed to diffuse in the tissue. So, and is degraded by some degradation rate here. What is the action of this signal? It has two actions. The first one is to trigger the fiber insemination. So whether there is signal, you can imagine that as a chemical that says, okay, we need to rebuild some network here. So fibers will inseminate, and it blocks the differentiation of adipocyte cells. This enables to just delay the apparition of the cells, as we know from that real system do. In the proliferation phase, it's just the reconstruction of a matrix, and then the adipocyte cells come after. So this is what this chemical is trying to do. So with this rule, you cut. That means that you create gradient at the two borders of your injury. That triggers the formation of fiber insemination. So you move the border. There is still a gradient, so there is still chemical. So the two borders will move towards one another, up until you fill the whole injury zone with some matrix. So then the chemical do not create anymore because there are no gradients. So it stops being created and degrades with the degradation rate. So at a point you have no chemical, and then the cell can be inseminated again in your matrix. But depending on the connectivity of your matrix, then cell will be able to appear or not. And this enables to have three different architecture. You have a case where what you will build would be a very highly connected, highly dense matrix of fibers that do not enable to bring you adipocytes, and you will have some regenerative repair. So here it's an illustration of the outcomes that you can have with this model, this new mechanism. After an injury, depending on your model parameters, we observed that we can rebuild the tissue where the injury is filled with highly connected and dense fiber network with few cells. We have tissues where we reconstruct something that at least visually is very close to what we had before injury, that we call regenerative repair. And we have something in the middle. We have some injuries where we have some matrix, we have some cell. It's not exactly reconstructed, but it's not a fibrosis either. So we call it unstructured tissue. So the question, these are the three possibilities that the model offers. The question is which parameter drives what outcome. So in a first theoretical work analysis of this model, we did by hand sensitivity analysis of the parameters, and it enabled to detect the number of cross-link in the matrix as an essential parameter driving tissue outcome. What we observed with this with the analysis of the model was that regeneration or scar formation could be controlled by the number of crosslinks in the newly created fiber here. So more precisely, what we did was to use our morphological quantifiers before injury and after injury. And this enables to compute what we call a regenerative score. So this quantity here, that is basically just the distance between our structures after reconstruction and the structures before. So the smaller this quantity is, the more regenerative, the closer our structures are before and after. And so if we plot this quantity as function of PF, so this is linked to the number of fibers in our ECM, we see that there are some optimal values that lead to regeneration, and if we increase the number of cross-link, then we will create a scar. So this was first set of interesting results that already pointed to the fact that the connectivity of the matrix can be a major driver in the outcome of regeneration. And so we wanted to test this prediction, whether we could indeed in vivo induce a regeneration by playing on the mechanical properties of the matrix. So it leads us to the last part of this intervention is it led us to envision a combined in vivo and in silico approach to interface the prediction of the model to some experiments in mice. We knew already, and you know since Jenny introduction, that regeneration is possible in mice. As Jenny mentioned initially, the works in 2018 of Elodie Labitte and co-authors in Restore already showed that it was possible to induce regeneration in adipose tissue of a mature mouse by injection of opioid NALM, opioid receptor antagonist. It is not completely reasonable to do that in animals or in humans, because this is associated with a lot of pain. So there is an interest of finding another therapeutic target that is less pain-inducing to induce regeneration. But at least what it shows is that the system, so the body, has the ability to regenerate. It's just that it did not select that during evolution. It selected the wound healing outcome, but it can. So there is hope to be able to induce it by some other factors. What the model suggested so far was that if we find a way to play on the mechanical properties of the INCM matrix that is produced after injury, then there is a possibility to induce a regenerative repair. So this is a scheme that represents the work that we did in this combined vivo. So we interfaced in silico experiments with in vivo experiments here. This was done in 4 steps. The first one was a calibration of the model on experimental data. Then we did an exhaustive analysis to quantify really the role of each parameter to be sure that, to identify really the main parameters driving the outcomes. And then we looked at how we could modulate in silico this parameter, whether we had to play on it during a long time or whether we could play only in a windows of time to induce regeneration. And then we tested this prediction in the mice model experimental. So the first step was to transform our models, so the morphogenesis and the repair models, into a user-friendly interface that was distributed at the Restore lab, and that was used by the PhD of Anastasia Paccari, who worked on it during 3 years. So this is a software tool that enables biologists to do in silico experiments. So you have a part where you can generate simulations of morphogenesis by playing on the parameters that you know already now. You can choose the length of the fiber, the number of fiber, the number of cells, the connectivity of your matrix, the remodeling speed, etc. And then you can send the simulation, visualize it, and automatically compute the morphological quantifiers, such that the elongation of the cell cluster, the alignment of the network, etc. So this is the left part of the interface. And then on the right part, you can do experiments of tissue reconstruction. It means that you can cut part of your tissue and then send a simulation of repair. Again, playing on some parameters, so you can control the frequency of apparition of new fibers, the cross-linking of this new matrix, and the unlinking of this new matrix, for instance. So what did this interface enable is that it enabled to perform a lot of simulation. I think Anastasia did not far from 2,000 or 3,000 of different simulation, with exploring 400 combination of values of the parameter. The goal here was to provide a multi-parametric analysis of the model in order to identify precisely which parameter did what on the outcome. So of course, if you have 3,000 simulations, you have to have a way to analyze them systematically. So, and it's really hard to do it by yourself, and especially if your region of the dimension of your parameter domain is more than 2 or 3D, because with your brain, we are just limited to our 3D vision maximum. So imagine that you have 10 parameters at the same time. It's impossible to analyze directly. And this is where machine learning can help. And for me, it's really where it has its interest and not in anything else. But it can help to analyze systematically large quantities of data in high dimensions. This is what we did here. So we had this pool of 2,218 simulations, and each of them was associated with a score of repair. So it was located in one of the 3 cases, in one of the 3 possibilities, the regeneration part, the unstructured tissue, or the scar tissue. So each simulation has one of the box, and so we could build a machine learning model that was trained on the data to recognize which issue, regeneration, unstructured, or scar, it would put given the parameters that were given at the entry. Okay, so it was trained on some part of the simulation and then tested on the other part of the simulation. So he learned to recognize what was the issue as function if you feed it some parameters. The idea of having this neural network that is trained on your data is that afterwards you can pass an explicability algorithm to analyze how your neural network learned to recognize one issue or the other. This is based on, so the name is a SHAP value. It's based on a game theoretical approach that consider the explanation of your network as a game, where the parameter values are the players. And what it enables you to know is the influence of each player on the outcome. So this is an explicability tool that you pass after the building of a neural network that enables to detect which parameter had the most influence in the prediction of your neural network. This is a very powerful tool because this is the main problem of neural networks, is that we don't know how they learn. We don't know how they decided how they decided to predict an outcome. There's a way of inverting the neural network. And so this SHAP value actually revealed that the parameter that had the most influence on the prediction of the neural network was the linking parameter here. The second one was the unlinking, and the third one was the density, so the number of fibers. So that means that in order for him to learn which which simulation would end up in the regeneration class, he needed mostly to know the linking of the matrix, the linking parameter. And basically for the regenerative class, he would learn that with higher probability, you would end up in the regenerative repair class if the linking of the matrix was low. And with higher probability, you would be scarred in the scar, wound healing class, if the linking was high. So in any case, in all the parameters tested, the three first ones were the ones concerning the matrix, and the two first were the one concerning the cross-linking of the matrix. So that was giving some quantitative proof that the number of cross-link in the matrix can indeed be the main parameters driving regeneration or scar formation. Okay, so this explainability enabled to detect this parameter as this quantity as major in the outcome. And then we asked whether, so now imagine that you want to test that in vivo. You have to find a molecule that enables to modulate actually the connectivity of your matrix. Actually, it exists. There are several of them. And the question is, how do you, if you want to test it, you want to decrease your number of cross-link, then the natural question is, okay, I do an injury in my mouse, then I inject the product, but in which temporal window do I inject the product? Do I have to do it every day, for how long, etc.? So we questioned that in the in silico model, whether there was a temporal window that was sufficient to induce generative class by playing on the cross-link parameter. So in silico, what we observed, if we plot the number of fiber links as function of time after injury here, what we observed was that we had a transient evolution of the number of cross-linking. And that in any case, even in the scar repair class, so when the crosslink of the matrix is not modified, it's quite large, then we saw that the number of links was increasing in the 5 days post-injury with the model, with the simulation. And it was the same also with the regenerative repair. You see that it increased even a bit more in the 3 first days. So that suggested that if we find a way to decrease the number of crosslink in the 5 days post-injury, we could already change drastically the architecture. So in silico, we did that. We took a simulation that with its normal classical linking would lead to a scar repair. And we modified the parameter transiently for 5 days after injury. We decreased, we played on the connectivity of the matrix to control it, to keep it low, at least for 5 first days. And then we looked, and indeed, we were able to pass to a regenerative repair class. That suggested that playing on the matrix crosslink can change the outcome, and you only need to play it in the 5 first days. This is really the most important time window in which you should act. If you play afterwards, it's too late. So the model really predicted the 5 days. And so this experimental protocol, was put in real tissue, in real mice. So did exactly the same thing. If you cut part of the adipose tissue of a mice and you let it evolve, it would converge to a scar. And so the biologist introduced this BAPN molecule that is an inhibitor of the lysyl oxidase, that is a molecule that favors the crosslink of the collagen matrix. Sorry. For the 5 first day after injury. And indeed, what they could observe is that the tissue started to rebuild in a way similar to its architecture before injury. So there are several quantifiers that enable to quantify the regenerative stuff. So when we say regeneration, be careful that we are talking about architecture. We're just asking whether the tissue that is reconstructed looks the same as before. Looks the same in quantitatively. I mean, in the simulation, you remember that we compare the number of cells and the morphology of the clusters before and after. Experimentally, look basically at some light quantifiers that are based on light that can detect the presence of cells or not. And we saw that with the experiments with BAPN, we reproduced indeed a tissue where adipocyte cells reappeared. So you see that it's reconstructed, while in the control condition, we see that it's only composed of a highly cross-linked matrix with no differentiated cells. So this is where we quantified here the regeneration of the tissue. Okay, so actually this study combined in silico in vivo study and ended to convince us that the model, this very naive and simple model, was actually a very powerful tool that had a prediction abilities, because it enabled not only to detect a new therapeutic target to play on, to induce regeneration instead of scar formation after injury, but it also enabled to predict a time window of 5 days that was sufficient to induce this change in outcome. So that gives solid basis to the development of a digital twin. What it means to be qualified as a digital twin is the sense that it cannot be adapted on live, like in real time, to some other tissue. You have to recalibrate your model. If you have a different system, a different tissue, then you have to recalibrate your parameters by comparing. It cannot adapt directly. So there is this real-time window that is a bit different than a digital twin, but at least it's a predictive tool because it enabled us to study new mechanism and predict some new results. So of course, there are a lot of, the other thing that is very interesting and surprising with this model is that already in 2D, it has this predictive power. We are sure that before going to 3D, the model was not able to give insights into the real system because it was too simplified. Actually, it showed that the mechanisms that we put are general enough to be able to be already very precise and sufficient for the real system. But of course, if you want to change scale and go towards a whole organism, there is the need to develop 3D, this model in 3D. And especially because we are interested in segregation in between two populations of very different elements, right? The question that we have, we know in 2D already that if we put spherical cells into anisotropic fiber network, more or less cross-linked, we create cluster of cells inside a more or less aligned network. Okay, this is what we had in 2D. The question is whether it's true in 3D, because for instance, for a network of fibers, it's way more simple in 2D, in a plane, to create lines of fibers that will surround stem cells. In 3D, because of the third dimension, and so the whole new possibilities of orientation, it's way less clear that structures will be able to close, fiber structures will be able to close around the cells. So we needed to question whether we would have lobules also in 3D. That was not obvious. And so that was the PhD of Pauline Chassonnery that extended in the most natural way the rules that we put in 2D into 3D. So the disks of the cells were, of course, extended to spheres. This is really easy for the cells. And for the fibers, we consider spherocylinders. So here, this is an image of the fiber network, but the spherocylinders are not represented. Here, they are represented only by arrows of their center. But they have a body, so they are really spherocylinders, and they can connect to the ones that they cross. And she was able to show that in 3D also, we could have the spontaneous emergence of clusters of cells surrounded by fibers. So that showed that actually this model was enough also in 3D to produce at least this segregation of cells in architectures that are close to the ones that we observe in real systems. So okay, so this is so the question is that in 3D, is it still true that we can induce change the regenerative or the outcome of reparation if we play on the matrix? There are good chances that that it could work as in 2D, at least for the morphogenesis part. It does. Another perspective of this model, as you probably notice, is that the biological rules that we put. So we are in the development phase, like from the establishment of the adult tissue, let's say, because we impose the number of cells that we inseminate, and we impose also their maximal size. So in this naive model, cells just grow with some law that is prescribed and up until a maximum size. So this model is not relevant, for instance, to question the role of different nutrients during life, right? Our tissue will just evolve and then it would stop because everyone is at maximal size. So one natural perspective of this work, and because we have in mind aging and obesity, is whether we could combine this mechanical model with some energetical model that takes into account the metabolism. That means that we would need to change our growth laws, so the laws of apparition of cells and growth of cells, as function of an external nutrient that represents actually the energy, so the resource, for instance, that is given from the organism to the adipose compartment. And this resource can be negative, because if the body is starving, for instance, it will need some energy from the tissue, so it would make cell decrease. So we need to modify our growth flow to take into account the energy deprivation of the organism and the nutrient-dependent growth flows that we don't take into account for the moment. This is actually a work in progress in the frame of a national project that we started last year. There are a lot of other questions. I invite you to think of all the different stuff that we could do, because it's really a simple modeling, simple model. Okay, so to conclude globally on here, before passing to the questions, so I always leave some time for discussion afterwards. Basically, what this long study, so we present that in like 2 hours, but it's a 9-year story, right? So from the beginning of our morphogenesis problem. As Jenny mentioned, this really long work that involves a lot of different developments. But it's a powerful approach, at least for me. It's one of the success story where mathematics is really used as prediction. It created a simplified framework, but proposed, enabled to build a powerful tool that enable to explore hypothesis on real system. Okay, in the general conclusion, so of course, all these studies necessitate to build relevant mathematical models. So you have the question of calibration of your parameters and validation of your model. So, and to validate, you have to compare the result of your model to things that you explore in the real system. And this is accompanied by a lot of challenges in the real system. Like, for instance, if you question architecture, you need to have segmentation tools, you need to define correct quantifiers, morphological quantifiers, etc. And then you have to do this going back and forth between model and data in order to validate and select the correct parameter space that enables to have this quantitative comparison. So there are a lot of things that are behind. You saw I mentioned a bit some tools from machine learning to help in the sensitivity analysis. This is also a huge part of the work that took some years that took a whole PhD actually. So yes, every step necessitates a lot of discussion and a lot of different tools. So mathematical theories, numerical studies, machine learning pipeline, etc. But in the end, it enables to have a system, for instance, that can model the years of development of a tissue in a few hours of computation. So we go back to the fact that the mathematical model enables to go far in time and in a most efficient way compared to experiments or observation on real tissue. Okay, so I hope we convinced you that it was not completely crazy to use mathematical framework to study real system. Now, yeah, maybe we can have a discussion or place for some questions. Thanks for your attention.
Another argument for using a mathematical modeling is also, I think most of you know that experiment on animals is more and more severe. So we are pushed to find an alternative way also to experiment and so to minimize animal experimentation. And as yeah, I told you, we perform more than 2,000 simulations in silico, so use that you cannot do in mice, so for sure. So you can try it with yes. And it was really helpful. This interface was really helpful because for us, we just have to change the number of parameters. We don't have to coordinate. That's it. So it was really convenient. And we can see what happened at the end. So mostly it's observation after it's all analysis with machine learning. So I don't know if you have any questions. If there is a lot of math, not choose to do math. Yeah, okay. Or you can send me an email at my email address. I can send you the presentation. So yeah, we have your email, Anjan. Yeah. So you can see it. Now we saw your
Yeah, sorry. It's a reflex, sorry. Always cleaning.
Yeah. Yeah. And sometimes also with mathematical, it's not what happened clearly in vivo because we put degradation, and so the remove of unlinking is not true in vivo. There is no enzyme or molecular process that removes the cross-link, but the result at the end is the release of mechanical pressure and the mechanical problem. So here the unlinking is just to mimic this release of mechanical constraints for the tissue, and there is no really degradation of the matrix. So sometimes there is some assumption that you cannot, yeah, it's not true in the real world, but the result at the end is the same. I don't know if you have any question. Okay, so that's it. I don't know if you have.
I have a question.
Yeah.
For the exam, for the article presentation, what we are required to do exactly?
I will send you the requirement, but for you, it's just to know what is the model used, what is first the biological question, also all the assumptions. Yeah, so you cannot do the all tissue. What the models that he used in this article, and why this model, in fact, because it's different kind of model, so you will know more at the end of this week. and also the result, and also to see the limitation also of the models.
Okay, are we required to learn the equations? Just
No. It's just for you to understand what is mathematical modeling. I'm not a biologist too, so I don't go through astro equations, so sorry. But to know the overview of this question and why we use this.
Yeah, no, of course, the point was not for you to be able to write a reaction diffusion equation at the end of this course, right? It's just at a point, there is something for the different interventions that I did with biologists, is that we hide our equations. we don't show them, we just show numerical simulation, the results, etc. Well, I think it's a mistake that we do. The problem that we have, so mathematical biology, you have to keep in mind that it's quite a recent field in the history of science compared, for instance, to mathematics for physics. It was because for a long time, biology was considered too complex to be prone to simplified visions offered by a mathematical model. And you see that we just simplify too much the system. In our example is the first one where the tissue is reduced to some cells and some segments. It's not even cells and fibers, it's disc and segments. So it might appear crazy to use this mechanical, naive stuff. But actually, the point is that we can still use this framework to identify mechanism. It just does not mean that the tissue is composed only of that. It just means that this has this influence here. And so it's accompanied with equations. So now I prefer at least to show them, just to say, okay, there are different mathematical methods, different mathematical models, and it's important also just to get used to see them, and just to see that there is an object. It's a function, actually. And what we call density is a function. What we call agent-based model is something that describes the position of elements in time, etc., etc. But it's not that you should recognize directly or whatever, just to show you 1 time, 2 times, etc. And at the end, you would be used to recognize which objects you see. So yeah, don't worry, we won't ask you to derive a continuum model from agent-based formalism. Even mathematicians don't know how to do it correctly. So.
I don't know if you have any questions. Okay, so you're free. But if you want to have question also in French, or hesitate afterwards.
Thank you.
Thank you.